Shall I start, sir? Good evening, students. Today we are going to discuss about Unit 6, Culture of Pearl, that is from MSc second year. So in this chapter, what we are going to discuss general description of mollusks and pearl mussels, systematic position of Indian pearl mussels, their general morphology, shell structure, gross anatomy and functional aspects, digestive system, circulatory system, respiratory system, excretory system, nervous system, and reproduction of freshwater mussels. Other than this, we are going to discuss about various culture practices, freshwater culture practices. In that, we are going to discuss about collection of mussels, pre-operative conditioning, surgery or implantation methods, mantle cavity insertion, gonadal implantation, pond culture, pond requirement, pond and feeding, etc. So let us start with the topic, students. So pearl is counted among the 9 gems and needs no cutting or polishing to bring out its luster. So most jewelry is fashioned out of precious metals and jewels that are found buried in the earth. But pearls are found inside a living creature, that is an oyster. Most pearls are produced by oysters in both freshwater and saltwater environments. Now, what are these oysters? You can see in the picture clearly how they look. And these are bivalves. There are 2 halves, so we can call them as bivalves. So oysters are bivalves, which means that its shell is made of 2 parts, or we can also call them as valves. Now the shell's valves are held together by an elastic ligament. This ligament is positioned where the valves come together and usually keeps the valves open so the oyster can eat. Now, how is a pearl formed? So these pearls are made by clams, oysters, and mussels. When a grain of sand or other small irritant gets painfully stuck in the mantle of the creature, that means if there is a sand grain or any microorganism enters into the body of this mussel, it gets irritated. So to avoid that irritation, what it does, like how when something falls in our eyes start watering. That is the mechanism, the protecting mechanism of our eyes to send that sand grain outside. Same way, the muscles also, they secrete a certain chemical so that it forms a layer around it so that it won't get affected. So a grain of sand or other small irritant gets painfully stuck in the mantle of the creature. The animal coats the irritant with the same material to make the shell. So this makes the irritant smooth and less painful. So when there is a layer formed around that same sand grain, that muscle won't get that pain anymore. So that is how over a period of time, these layers, layer by layer around that sand grain or a microorganism or any creature, any small creature. So it forms like a pearl. Now, these pearls are freshwater mussels. They belong to a phylum called as mollusca. Now, let us see the characteristic feature, the general characteristic features of mollusks. So mollusks are soft-bodied, bilaterally symmetrical, segmented, coelomate animals, usually shelled, having a mantle, ventral foot, anterior head, and a dorsal visceral mass. They're essentially aquatic, mostly marine. So few freshwater and somewhat terrestrial form also. They may be found as hidden parasites in the interior of other animals at times. They vary in size from giant squids and clams to little snails or a millimeter long. So these are the general characteristic features of phylum Mollusca. Now, if you see the types of pearls, what we come across are they can be natural pearls, cultured pearls, or artificial pearls. So now how natural pearls are prepared, how cultured pearls are manufactured or prepared, we'll see in detail. If you look at freshwater mussel, it belongs to phylum Mollusca and class Bivalvia, order Unionoidea, family Unionidae, and genus Lamellidens, species Marginalis. So we are going to discuss particularly about this one species called as Lamellidens marginalis. So these are commonly known as mussels, belong to phylum Mollusca. So class, as we said, Bivalvia, and subclass Paleoheterodonta. So they are freshwater bivalves of India. They usually burrow in the mud at the bottom of the pond. Inside the shell, a delicate layer called mantle envelops the whole visceral mass. The mantle comprises of two lateral halves called mantle lobes. The mantle lobe at the aboral side produces two short tubes, the inhalant and exhalant siphons. Water comes in through the inhalant siphon, carrying food and oxygen, and goes out by the exhalant siphon carrying excretory products. You can look into this picture where exhalant siphon is there and where inhalant siphon are there. So these are the external features of a freshwater mussel. Now these mussels, they are thick and cylindrical, run transversely across the body from one wall to the other, and they are responsible for the closure of the shell also. So this is exhalant siphon, posterior abductor muscle, heart, the gills, labia pulp, visceral mass, anterior abductor muscle, and the foot, mantle, and inhalant siphon. So these are the parts of this freshwater mussel. Now, how that microscopic structure of shell looks like. In section, the shell has three layers: an outer brown, horny layer, the periostratum, which is protective and is made of a horny organic material called conchiolin. So below it, the middle layer is a thick prismatic layer made of vertical crystals or prisms of calcium carbonate separated by conchiolin. The innermost nacreous layer of the mother of pearl layer is made of alternate layers of CaCO3 and conchiolin. The hinge ligament is made of uncalcified conchiolin. It is continuous with the periostracum. Reserve calcium carbonate for the two inner layers of the shell is stored in certain cells of the digestive gland. The nacreous layer is thickest at the umbo and the thinnest at the shell margin. It is used for manufacturing buttons. Now let us discuss in detail about the various systems of this freshwater mussel. Now, how does this alimentary canal work? So the digestive system of freshwater mussel, the first one we are going to discuss about alimentary canal. Under that, the first part is mouth. So immediately below the anterior abductor muscles lies the mouth, which is a transverse slit. On each side of the mouth are two ciliated triangular flaps, the external and internal labial palps. The external palps unite in front of the mouth to form an upper lip and the internal palps. They unite behind the mouth to form a lower lip. Now the palps bear cilia. Now esophagus. A short tube runs dorsally from the mouth to open into the stomach. If you look into the stomach, it's a large oval sac embedded in the digestive gland. The epithelium is thrown into folds. Two narrow ducts from the digestive glands open in the stomach. Occasionally, a mucus rod, the crystalline style, which plays an important role in digestion, is found in the stomach. It also contains diastatic enzymes. Next, intestine. The intestine is a narrow tube, and after its origin from the ventral surface of the stomach, near its posterior end, descends into the visceral mass above the foot and form two incomplete loops, ascends to the level of the stomach, anterior to the pericardium, turns sharply backward and proceeds straight as the rectum. If you see into rectum, the rectum pierces the pericardium and the ventricle of the heart, which runs parallel to the dorsal surface of the muscle and opens into the excellent siphon by the anus. The internal wall of the rectum is thrown into longitudinal folds to form a ridge known as typhlosole. Digestive glands. The digestive glands are a pair of irregular light gray structures situated around the stomach. The secretion of the glands is discharged into the stomach through small ducts. Each gland is made up of numerous diverticula. Now, the feeding of the freshwater mussel. The freshwater mussel is a ciliary feeder, that is, it collects food with the help of cilia. The food consists of detritus, that is, partially decomposed organic particles suspended in water, and microorganisms like bacteria, protozoa, etc. Detritus constitutes 4/5 of the total amount of the food. The respiratory water current created by the movement of cilia on the gills carries food particles and other matters. The heavy particles drop in the mantle cavity and are expelled out through excellent siphon by the movement of cilia in the mantle. The gill laminae are covered with a thin sheet of mucus and the food particles are entangled in it. The movement of the lateral cilia on the gills drives mucus sheet to the ventral edge of the gills bearing frontal cilia. The frontal cilia drives the mucus string forward to the ciliary tracts of the labial palps. Further assorting takes place here. The larger particles are carried to the tips of the labial palps, drop off in the mantle cavity and removed to the exterior. The mucus string with the food matters are carried to the deep groove, the food groove between the two palps, leading to the mouth and driven to the esophagus. Let us look at the diagram of the digestive system of freshwater mussel. This is called the foot, the structure foot. Then this is the mouth, anterior abductor muscle, stomach, digestive gland. This is all dotted area, digestive gland, pericardium, ventricle, auricle, and this is rectum. So this is the heart which has auricle and ventricle and it is covered with the pericardium and rectum and anus. This is the digestive system of lamellidens. Now let us see into the respiratory system of lamellidens. The respiratory organs consist of a pair of gills, one on each side between the body and the mantle. These are the interlamellar junction. This is the water tube. These are the branchial filaments, ostia. So each gill consists of two laminae, the outer and the inner. And each laminae consists of two lamellae, outer and inner. The two lamella of each lamina are united along the anterior, ventral and posterior edges, but free dorsally. The cavity inside the lamina is divided into several chambers or water tubes by means of vertical interlamellar junctions. So each lamella is made up of many gill filaments connected by interfilamentar junctions. Minute pores or ostia are present between the filaments. The outer lamella of the outer lamina is attached to the mantle. The inner lamella of the outer and the outer lamella of the inner lamina are attached together to the visceral mass. The inner lamella of the inner lamina is attached to the visceral mass. So all water tubes of the gill open dorsally into a suprabranchial chamber, which in turn opens to the exterior through the excellent siphon. The cells of the gill filaments are provided with cilia, and by their action, a current of water inhalant current enters the mantle cavity through the inhalant siphon. From there, it passes to the water tubes through ostia and goes to the suprabranchial chamber to pass out through the exhalant siphon as exhalant water current. The gill filaments are highly vascular and gaseous exchange takes place there. Heavier particles carried by the inhalant water current drop in the mantle cavity. Special cleansing cilia sweep small non-food particles off the gill filaments. They are entangled with the mucus secreted by hypobranchial glands in the root of the mantle cavity. The particles in the mantle cavity are swept to the exterior with excellent water current. The mantle is richly supplied with hemolymph and gaseous exchange occurs there also. Now let us look into circulatory system of lamellidans. The circulatory system is of open type. The contractile heart is enclosed in pericardium and has a median ventricle and two auricles. From the ventricle arises the anterior and posterior aorta, which supply oxygenated blood to different parts of the body through an extensive blood spaces or sinuses. So this is the pericardial layer, where this is the ventricle. So the anterior and posterior aorta join the ventricle, while the auricles are connected to gills and mantle lobes by branchial veins. The oxygenated blood from the gills and mantle passes into auricles and reaches the anterior or posterior parts of the animal through the ventricle. The blood from the different regions is carried by the vena cava directly to the mantle and gills or through the kidney for purification. The blood of the freshwater mussels is colorless and contains leukocytes. If you see into the excretory system of lamellidans, there are a pair of kidneys or organs of Bojanus and a pericardial gland or Cabus organ constitute the excretory system. Kidney. The kidneys are more or less U-shaped tubes located one on each side of the visceral mass, ventral to the pericardium. The brownish, spongy and glandular ventral arm is kidney proper, while the thin-walled, non-glandular ciliated dorsal arm is the urinary bladder. The cilia produce an outward current. The kidney proper opens in the pericardium through a renopericardial aperture, and the bladder communicates anteriorly with its fellow of the other side by a large oval aperture. Each bladder opens in the mantle cavity through a minute renal aperture between the visceral mass and the inner gill lamina of the side. Pericardial gland of lamellidans. It is a reddish-brown glandular mass just in front of the pericardium and discharges the excretory matters in the pericardial cavity. If you look into the nervous system of lamellidans, it has cerebral ganglia, cerebral commissure, and pedal ganglia. What is cerebral ganglia? Two triangular in shape, yellow in color, located a distance from each other, lying at the base of the labial palps, just under the skin. Cerebral commissure, a slender nerve, runs around the esophagus in the anterior part of it and connects the two cerebral ganglia at their anterodorsal border. Pedal ganglia. The pedal ganglia are two embedded deeply in the muscles of the foot at about 1 centimeter above the ventral border of the foot. The ganglia are oval in shape, pale yellow in color, and the two fuse medially to form a bilobed mass. Cerebropedal connectives. Arising from the posterior border of cerebral ganglia, the two nerves run downward, backward through the visceral mass and muscles of the foot to join the pedal ganglia at the dorsal border. Coming to visceral ganglia, two large ganglia partially fused and located ventral to the posterior uptake muscle, covered by a thin peritoneum. The ganglia are flattened, triangular in shape, and pale yellow in color. The two in vivo assumes X-shape, cerebrovisceral connectives. Two arising from the posterolateral border of cerebroganglion. Each runs backward, ventral to the attachment of the gill lamina with the visceral mass. It courses through the mass of the kidney and emerging from its posterior border runs further backwards to join the visceral ganglion of the side at its anterior border. Next, coming to the reproductive system in lamellidans. The sexes are separate, gonads are paired, and occupy the major portion of the visceral mass in which the loops of the intestine are lodged. Both the testes and ovaries are racemose glands. The gonoducts arising from the gonads are short, narrow band open the front of the renal aperture through a round genital pore. Male reproductive system consists of testes, which are white in color, and a vast difference arises from the posterolateral border of each testis. Female reproductive system consists of ovaries, which are reddish in color, and oviduct, which arises from the posterolateral border of each ovary. This is the reproductive system of lammellidan species, which has mouth. This is the gonad, intestine, digestive gland, stomach, and this is the gonoduct. This funnel-shaped structure is the gonoduct, and this is the layer around the heart, which is pericardium, and this is rectum, and this U-shape, this structure is called as the kidney, and then this end of the tube is the anus. Now let us see the fertilization and development in lamellidans. The fertilization is internal. The eggs discharged in the mantle cavity from the ovaries through female genital pores pass into the suprabranchial chamber. The sperms discharged from the testis in the mantle cavity through male gonopores pass out with water current and reach the suprabranchial chamber of the female. This fertilization takes place there and the fertilized eggs are passed to the water tubes. In the water tubes, a number of fertilized eggs are enclosed in a thick, soft, somewhat triangular whitish capsule, which is called as the egg capsule. So development continues in the egg capsule and the capsules with the glochidium larvae are discharged outside. The glochidium larvae escape from the capsule. So if you look at this picture, you would know how the glochidium larva looks like. It has hooks, it has byssus, the sense organs, shell, and the abductor muscle. So it has presence of a bivalve shell, valves united dorsally and flat ventrally. Ventral ends of the valves curve and bear spines. Mantle lines are walls of the shell and bears brush-like hairs. An abductor muscle at the base of the shell connects the walls. Body is an undivided mass. Presence of a byssus gland near abductor muscle. And then a long byssus thread present. So this is the long byssus thread. Now, let us discuss about metamorphosis. What is the meaning of metamorphosis? So the larva, when it undergoes so many changes to become an adult, so the stages that it undergoes, it's called as metamorphosis. So the Glaucidium or the Glaucidium larva attaches itself to the gill of a freshwater fish by the byssus thread. The larva penetrates the host tissue, becomes enclosed by gill epithelium, live temporarily as ectoparasites, and undergoes metamorphosis. The byssus thread and sense organs disappear. A stomodium is found by ectodermal invagination immediately posterior to the roof of the byssus thread and soon communicates with the archenteron. The opening of the stomodium forms the mouth. Now, proctodium is not formed. The posterior end of the archenteron is in contact with the ectoderm and the anus is formed by a simple process of rupture. Food originates as a median ventral elevation behind the mouth. Gill rudiments appear as two papillae on each side of the mouth. The larvae fit for free life, drops from the host, sinks to the bottom and gradually attains adult form. Please look at the picture of this flowchart which shows the life cycle of a mussel. So this is the juvenile and gets converted into the adult and it gives out the Glaucidia larva, which in turn grows into an adult one. So this Glaucidia, it affects the it gets attached to the gills of the fish. This acts like a host now, fish host. Now we have seen all the systems of this lamellidin species. Now let us get into the pearl culture. So a cultured pearl is a pearl initiated by a mussel or an oyster farmer under controlled conditions using two very different groups of bivalve mollusks, the freshwater river mussels and saltwater pearl oysters. Now, how does this process start? The process of pearl culture. The process of pearl culture includes the following steps, which are very crucial for obtaining high grade of pearls with good commercial value. Step 1, construction of pearl form. So it includes 3 steps. They are selection of form site, construction of form, well-planned work schedule. These steps determines the type of pearls produced and the oyster survival rate. Some of the points to be noted while selecting the site are natural features like mountains and reefs are needed to protect the farm from winds, currents, storms, etc. Constant regularity of temperature, types of seabed such as rocky or sandy. Gentle currents are essential for the survival of the oyster as they bring food and oxygen. If you look at the first step, that is site selection, the selected site for pearl culture should have a pollution-free water supply and no algal blooms. That means it should be free of any microcysts. Water should be clean and with low turbidity, as highly turbid water reduces the filtration rate of mussels. Obtaining pearl mussel stocks, as mussels take 6 to 10 years to attain implantable size, the mussel stocks for culture or implantation procedures are generally collected from natural water bodies like ponds or rivers. Mussels are handpicked from the pond bottom, and are selected on the basis of their size. Healthy mussels that have attained a length and weight of it's either more or less equal to 8 centimeters and more or less equal to 35 grams respectively, are selected for implantation in the pearl production process. Next is construction of pearl farm. The whole pearl farm system is based on the series of floating wooden rafts. 10 units of wooden rafts are used. Each raft consists of 2 to 5 pieces of wood, making the total length to 20 ft, that is 20 feet. The raft is covered with wire mesh baskets, each of which house 10 oysters. Well-planned work schedule. A typical work schedule plays a very critical role in pearl culture. The timing for collecting and seeding the oysters must be scheduled and followed strictly. Next is collecting oysters. After the construction of pearl farm, the divers set out to the bottom of the sea to collect the oysters. Divers are pulled by large lugger boats in the direction of the tidal flow. Then oysters are generally located on a flat rock bottom and are usually covered with marine animals and a thin layer of silt. Therefore, it is often very difficult for divers to recognize them. The shells collected are cleaned, sized, and placed into baskets for storage until they are transferred to the pearl farm. Next, coming to seeding. 2 to 3-year-old healthy oysters are considered for surgical implantation known as seeding. This is a very delicate operation and involves 3 stages. The first stage is preparation of the graft. A donor oyster is sacrificed to obtain mantle. Mantle is needed by the host oyster to accept the nucleus. The mantle is located on the outer section of the oyster and mantle produces the nacre which forms pearl. Before a graft is taken from the mantle, the oysters are starved for several days to slow down the metabolism of the oyster. This helps to decrease the risk of core rejection and open the oyster easily. Pre-grafting culture. Prior to surgical implantation, the selected muscles are kept crowded for 24 to 36 hours to ease the relaxation of abductor muscles. The muscles are stocked in FRP or ferrocement tanks in aged tap water at a density of 1 muscle per liter of water. Crowding of the muscles facilitates the smooth opening of the valves for implantation procedure. Now, before starting the surgical procedure, the muscles are kept in an upward facing position for half an hour, where the opening part, that is the ventral side, faces upward and the umbo, it faces the dorsal side. Now, how to insert that core particle? A nucleus is placed inside the scalpel slit and the oyster is then returned back to the water. The inserted core irritates the oyster, provoking it to gradually coat the core with thin layers of mother of pearl nacre. After some time, the oysters are collected and x-rayed to see whether the implants have been accepted or not. Oysters which have rejected the implant are returned to the water and are once again operated. The oysters which have accepted the implant are transferred to the pearl form. The person who is seeding must be extremely careful not to harm the tiny pea crab, which lives unharmed within every healthy oyster. So it is presumed that the crab assists the oyster by keeping it clean and by sharing the debris which the oyster sucks in. Next is grafting. Now, the crucial step in the entire pearl production protocol is the implantation of the nuclei or beads into the mussel. The nucleus made up of acrylic powder or shell powder can be grafted along with the mantle graft. That is a sliced mantle tissue taken from the edge of the mantle tissue near the pallial line that is obtained from the muscle that acts as the donor muscle. So that it later develops into a pearl sac. Now attaching the graft. So the oyster is opened with special wedges and pliers. There's a very sharp instruments that we can use those. Then a scalpel slit is made in the soft tissue near the reproductive organ. And a graft of living mantle is inserted into the slit. 3 methods of implantation are in practice. What are the 3 methods? Namely, first one is mantle cavity, mantle tissue implantation, gonadal implantation. So the choice of implantation method depends on the type of pearl targeted. That is, whether it is designer pearl, round pearl, or rice pearl, etc. Now, what is mantle cavity implantation? Out of the 3 methods, this is the simplest method requiring minimum skill and expertise. In this method, the nucleus is implanted into the cavity between the outer mantle layer and the inner surface of the mussel shell. Mantle grafts are not used in the mantle cavity implantation method as the outer mantle layer serves as the source of nacre secretion. Mantle tissue implantation. In this method, the nucleus along with the mantle graft is implanted into the pockets made on the posterior side in both left and right lobes of the mantle tissue of the recipient mussel. Next is gonadal implantation. Here, a small incision is made in the gonad of the recipient mussel, and then the nucleus together with the mantle graft is inserted into the incision. A live graft of 2 to 3 mm taken from the pallial mantle ribbon is inserted along with the round nucleus. So care should be taken to utilize the processed graft within 45 minutes to 1 hour. Otherwise, graft will be deteriorated and it may not be suitable for implantation. So implantation can be carried out throughout the year except May, June, that is the summer season, so as to prevent graft and mantle rejection as well as to minimize the post-operative mortality rate. The success rate of pearl production is 60 to 70% in the mantle cavity and mantle tissue implantation method, whereas in gonadal implantations, it is 25 to 30%. Now, post-grafting inspection. Following the implantation of nuclei, the muscles are subjected to post-implantation care for a period of 7 to 10 days to minimize post-implantation mortality and to minimize the rejection of implanted nuclei. During this time, the muscles are treated with broad-spectrum antibiotics to minimize rejection and for quick healing of the surgical process. They are kept for 24 hours and After a 3-day interval, again subsequently up to 9 days, and feed with green algae along with vigorous aeration. Then antibiotics from 1 to 2 ppm is added to the tanks stocked with implanted muscles immediately after the completion of the surgical procedures and after 24 hours of antibiotic exposure. So water exchange is carried out. Now post-grafting culture, that is the pearl development. Following post-operative care, the implanted muscles are shifted to the desired culture system, wherein they are kept in hanging condition packed in nylon net bags. The implanted muscles are cultured either in FRP, that is in ferrocement tanks, or ponds along with the other compatible fish species. The culture period of implanted muscles generally varies from 12 to 18 months, depending on the method of the implantation followed. The ambient soil and water quality parameters are conducive to the formation of good quality pearl in captive condition. Next is harvesting. The pond culture of operated mussels varies from 6 months or more, depending upon the size and number of nuclei implanted. The health of the mussels and the condition of the pond environment also. The culture units required periodic cleaning of accumulated silt, periphytic algae, and other fouling fauna finding entry into the units. At the end of the culture period, harvesting is done. The mussels are either crushed, followed by sieving to extract pearls, or the mussel is individually sacrificed, or individually pearls are taken out from the pearl sac of the live mussels without sacrificing. The later method, though difficult, is desirable to prevent depletion of stocks of mussels in the natural environment. So after 2 to 3 years, the oysters are harvested. It is necessary to make a trial harvest to determine whether the pearls have a sufficient coating or not. If it is not sufficient, then an additional 6 months to a year of culturing is necessary. The oysters are split open and pearl bags are cut by the scalpel to remove the pearls. Collected pearls should be thoroughly dried after the harvest to prevent loss of luster. Then, so the culture period in mantle tissue implanted muscle is generally from 12 months to 18 months also. The success rate in this procedure is about 60 to 70% of the muscles implanted. The culture period of the gonadal implanted muscle is generally 12 months. The muscles after the culture period are opened carefully and the position of the pearl is felt by touching the area close to the incision scar. By a pair of scissors, fine forceps, and needle, the pearl formed is carefully removed without cutting or damaging the intestine or other internal tissues. The muscle may also be sacrificed to remove a gonadal pearl. The layers of the gonads are cut open and the pearl is removed easily. Now, in this, normally what happens? We get different kinds of pearls. It can be regular, round, or unattached pearls are also obtained in this. So during sorting of pearls, so there are many different steps involved with the sorting of pearls. Firstly, the pearls are sorted according to whether they can be used for the culture pearl industry or not. Now, the pearls are categorized into 3 sections. So we have unmarked pearls, pearls with one major blemish, or pearls with more than one major blemish. We can see here the different colors of puzzles, light gray to dark gray, light pink, pale yellow, lustrous, silver color, ash color. So these are the different shades we can find in the pearls. The value of pearls by pearl type. We have different types of pearls in this picture. These are the freshwater pearls, Akoya pearls, Tahitian pearls, and these are South Sea pearls. So far we have discussed about all the various systems, digestive system, excretory system, reproductive system, and all the systems. So let me just brief it once again. So the Lamellidens marginalis, it belongs to the phylum Mollusca, class Bivalvia, order Unionoida, family Unionidae, and the genus Lamellidens, species Marginalis. So we have seen how the outer surface, how does it look? The internal organs, the microscopic structure of the shell, and the different parts of the digestive system also we have studied. What happens in the mouth, how does the esophagus look, and what is the function of the stomach. Then what is the function of the intestine, rectum, digestive glands, and the feeding pattern of these freshwater mussels, what is that they require to be fed so that in pearl culture, they grow properly without any damage. And the digestive system, respiratory system, under this also we have learned how the respiration process take place. And we have studied about the circulatory system of lamellidens. We also had looked into this excretory system of lamellidens, the different parts, how does a kidney function, how does the pericardial gland, how do they function? Then we learned about nervous system of lamellidens, in that the cerebral ganglia, cerebral commissure, pedal ganglia, visceral ganglia, and the cerebrovisceral connectives. These are the important organs that are present in the body of freshwater mussel. Then looking into the reproductive system, we studied about the male reproductive system and the female reproductive system, whereas male reproductive system consists of testes, which are white in color, a vast difference that arises from the posterior lateral border of each testis. Then the female reproductive system. These are the parts of the reproductive system: mouth, gonad, intestine, digestive gland, stomach, gonoduct, pericardium, rectum, kidney, and the anus. Then the how fertilization and development happens in the lamellidens, how the eggs get changed into the larvae. We learned about Glochidium larva, the presence of bivalve. Even this has two shells that are dorsally attached. Then valves are united dorsally and flat ventrally. Ventral ends of the valves curve and bear spines. These are the spine-like structures on either side. And then we also studied about metamorphosis, how a larva changes into an adult one. And then this is the life cycle of a mussel, freshwater mussel. So cultured pearls are the response of the shell on a tissue implant. A tiny piece of mantle tissue of a donor shell is transplanted into a recipient shell. Now this graft will form a pearl sac and the tissue will precipitate calcium carbonate into this pocket. So there are a number of options to produce cultured pearls. by using fresh water or seawater shells also. Now, the transplanting the graft into the mantle or into the gonad, and a spherical bead, or do it non-beaded also. These are the different structure procedures: pre-grafting culture, how to insert the core, and the grafting technique. Mantle cavity implantation, mantle tissue implantation, then gonadal implantation. Then post-grafting inspection after the surgery, what are the precautions to be taken, how they should be taken care, what type of food should be given, what type of antibiotics should be used so that the pearls, they don't spill the nucleus that we have inserted into that body. Then post-grafting culture, how the pearls develop. Then how to harvest the pearls. So hope you have understood, students. So in this, the frequently asked questions are: explain the digestive system of freshwater mussel, explain the circulatory system of freshwater mussel, explain the excretory system of the freshwater mussel, or explain the nervous system of the freshwater mussel, explain the reproductive system of freshwater mussel, or the various steps involved in the formation of pearl culture in the industry of pearl culture. What are the methods of grafting? Describe the technique of grafting, post-grafting or pre-surgical method. So all these types of questions will be asked, or maybe describe the anatomy of the freshwater mussel, etc. So with this, we are ending the session, students. Thank you.